Hi, everyone. Sorry for the delays. We were having problems with screen share and Crowdcast, which has been a repeating issue this morning, unfortunately, which is why we're running so late. But I'm pleased to say that we're back up and running now, and very happy to introduce our next speaker, who is Biyu Jade He. Dr. Biyu He is a member of the Neuroscience Institute and an assistant professor in the departments of neurology, neuroscience, and physiology and radiology at New York University, where she investigates perceptual processing in the human brain. Biyu completed her PhD in neuroscience from Washington University in St. Louis, and then subsequently went on to lead her own independent research group as part of the intramural early investigator research program at the NIH, National Institutes of Neurological Disorders and Stroke. She then joined NYU, where her laboratory uses a combination of invasive and non-invasive multimodal human brain imaging, brain stimulation, and computational approaches to investigate the neural mechanisms of perceptual processing in the human brain. She has also made original contributions to understanding the organization, mechanisms, and functional roles of spontaneous brain activity and aperiodic brain activity. For this work, she has received a number of prestigious awards, including, among others, a Society for Neuroscience Trubach Career Development Award, an NSF Career Award, and a Klingenstein-Simons Fellowship Award. And with that, I now hand it over to Dr. Biyu He. Please take it away. Oh, Dr. He, you're muted.
Oh, can you hear me now?
Yes.
Okay, great. Okay, thanks so much, Blake, for that introduction. I apologize that I had to upload the slides to Google Slides, so some of the fonts and pictures might be a little odd this morning. I like to start to say that I'm also a consciousness researcher, but today I'm going to show you data that's relevant to human perception, specifically neural mechanisms of human perception, including conscious perception. But I'm not going to have time in the talk to get into theoretical arguments about consciousness, but I'd be happy to discuss that afterwards. So there are two main tasks that any perceptual systems need to solve in the natural world. One is to infer about the cause of the current sensory input, and the second is to predict forward in time about the state of the future sensory input. For example, if you're swimming in the Everglades National Park and you see this in the water, the first thing you need to figure out is that a crocodile or a piece of driftwood. And then the second thing you need to figure out is that if it's a crocodile, is it coming at me? Should I get out of the water now? My lab has worked on both of these questions. Today, I'm not going to have time to talk about the second line of work, but you find papers on my website. I'm going to talk about a more foundational question, which is how does the brain perceive? And I'm not an AI researcher, but I hope that what I'll show you will be relevant to those who are interested in generating an artificial perceptual system that really captures human perception. So textbooks in neuroscience have taught us a lot about what happens in the feed-forward stream of sensory processing and perceptual processing. For example, we know that neurons at successive stages of the ventral visual stream extract more and more abstract and complex features from the sensory input. And this understanding of the feed-forward processing in the ventral stream is beautifully mirrored by the successes of deep convolutional neural nets, as we've heard yesterday from Jim DiCarlo's talk. So what we know from behavior and studies is that
so it only catches Allison.
I'm sorry.
I'm gonna write to her like we're running late.
I think there is a background.
She actually told me.
Pravesh, we can hear you. Please.
Okay.
So she said she's gonna be right on.
So what we know is that in the ventral stream, the invariance in neural processing builds up at higher and higher level of the hierarchy. And for example, higher order neurons in higher levels of the hierarchy have more invariant neural tuning to changes in low-level features of sensory input, such as changes in position, illumination, and pose. And this neural invariance directly underlies perceptual invariance, allowing us to do tasks such as identification and categorization, where sensory input can have very large differences, but nevertheless, we can identify an individual and categorize objects. But sensory processing doesn't just need to achieve perceptual invariance in the natural environment. It also needs to solve the ambiguity challenge. Natural scenes are often ambiguous due to clutter, shading, changes in illumination, and just inherent complexities of natural objects. And resolving ambiguity is crucial to survival. For example, our ancestors have to be able to recognize these hidden our camouflaged predators in the bush and try to look here, and then you'll see how difficult it is to recognize ambiguous stimulus in your visual periphery. So today I'd like to tell you about what we found about how the human perceptual system solves the ambiguity challenge. And at this meeting, we've seen some very nice work in the AI domain about how model systems computer models can solve ambiguity. So to solve ambiguity, what you need to do is to bring prior knowledge to constrain your perceptual interpretation, because sensory input is ambiguous. What that means is that it's compatible with multiple interpretations. So you have to bring prior knowledge to constrain perceptual interpretation. And perception is really the brain's model of the environment. We bring prior knowledge from lifelong experiences to constrain perception in a very automatic manner that the conscious volition cannot override. For example, you see, you see this as concave and this as convex. That's because you have the lifelong prior that light come from above, and you can't see it in a different way. This phase, I think the animation is not working very well here. Oh, sorry, because of the Google slide. Okay, I'll just say that this is a 2, 0, but if it's upside down, you'll see briefly a 3 instead of a 2 when it's upside down. And you know that you can't rotate a 2 to get a 3. It's because the sensory input is compatible with the letter 2, the digit 2 when it's upright, and with the digit 3 when it's upside down. And this knowledge about digits in your brain automatically shapes your perception. Okay, so so today I'm going to tell you about what we found about the neural mechanisms underlying prior knowledge that constrains perception. Specifically, about 3 different sources of prior knowledge, including past memories and spontaneous brain activity and top-down inference. So the overarching conclusive message here is that perception is the brain's inference about what's out there. And prior knowledge can include both latent memories as encoded in synaptic connectivity and active neurodynamics as embodied in constant fluctuations of spontaneous activity as well as top-down input. So first, let's dive into memories as a source of prior knowledge that shapes perception. About 50 years ago, the venerable British psychologist Richard Gregory created this picture that has instantly become foundational teaching materials in psychology. So if you've never seen this picture, what you see is black and white meaningless dots. But once you see a dog in this picture where the head is pointing down and the limbs are here, the body is here, once you see that, likely every time afterwards, when you are presented with this picture, you recognize a dog instantly. Interestingly, Dali worked this part of this picture into one of his paintings. And if you are in St. Petersburg in Florida, I highly recommend checking out the painting that he made. So we wanted to study in the lab how this sort of one-shot learning happens in the human brain. So we have created pictures like this. This picture probably looks like a meaningless pattern to many of you. And now if I show you this picture, now for a lot of you, you will see a dog out of this picture. And if this doesn't work for you, there are other examples that will work for you. For example, this one. And now many of you will see a cat out of this picture. It's a very dramatic example of one-shot learning. And just to show you the behavioral data that proves it, these are behavioral results from healthy human subjects, where if we present this black and white picture 6 times, you see the recognition rate is quite low. But a single experience of seeing this grayscale picture will boost people's recognition rate of the black and white picture substantially. And if this gray picture does not match the black and white picture, you see the repetition, mere repetition does not increase recognition rate very much. So it's a one-shot perceptual learning phenomenon, and it transfers by definition because yes, you see the grayscale picture and transfers to the related black and white picture. So until very recently, we didn't know how to classify one-shot perceptual learning in the broader scheme of memory systems. As many of you know, these pictures basically summarizes decades of neuroscience and psychology work on memory. We know that there are different memory systems. We know that the hippocampus-dependent memory system is very fast learning, and it encodes episodic memory, semantic memory, whereas it's typically thought that cortex and striatum dependent memory systems, such as motor skill learning, is very slow and deliberate. We also know that this is the declarative memory. It's called declarative because it's conscious memory that you can recall to the front of your mind, and on the other hand, non-declarative system memory is implicit and unconscious. So where do we place one-shot perceptual learning? It's fast, so it seems like it should be hippocampus dependent. So we tested that in collaboration with Larry Squire in patients with hippocampal lesions that gives them severe amnesia. This is a MRI scan of 5 patients. I'm sorry, the labels are shifted. This one is control. And the others are patients. Hippocampus is here. So in the first 4 patients, these first 4 brains, you see there are black holes here. Then hippocampal volume loss is about 8%, which implies about near complete total loss of neurons in the hippocampus. And this gentleman was very unlucky to have encephalitis that not only destroyed hippocampus, but also bilateral temporal lobe. Okay, so what do they do in this task? How do they do in this task? We found dramatically that patients with severe loss of episodic memory perform the task just as well as controls. This is the same behavior task that I showed you earlier. To the left, this is the black and white pictures identification accuracy before seeing the matching grayscale picture, and then these are the same pictures tested after seeing the grayscale picture at 1 day later, 7 days later, 5 months later. You can see that patients have this one-shot perceptual learning phenomenon, and it's long-lasting. And at 5 months, its patients are even better than controls. And the white dot here corresponds to this gentleman who had very severe memory loss. And it also showed that the beautiful dissociation between one-shot perceptual learning and one-shot episodic memory. Because if we ask patients to tell us what's the format of the task they performed, or to tell us if they have seen this picture before, they are severely impaired. But testing on this one-shot perceptual learning, transfer learning task, patients are completely intact. Okay, so this places one-shot perceptual learning in the branch of memory, the tree of memory systems under priming, which is cortex dependent. It does not depend on hippocampus, and it's one-shot and it's fast and it's transfers. So I think this is very important, has a lot of theoretical implications. So we wanted to know what is the brain mechanisms underlying this one-shot perceptual learning. So we combined MEG, which gives us whole brain coverage of neuronal dynamics at millisecond temporal resolution, with 70 fMRI, which gives us whole brain coverage with exquisite spatial resolution and sensitivity to cortical and subcortical regions to understand what happens. So we used RSA in this analysis to understand how each brain region represents a set of black and white pictures before and after seeing the corresponding grayscale picture. Basically, for any 2 images, for a particular brain region, we can look at its activity pattern. Then we can compute the neural distance between these two images. And then we can build this RDM, which tells us the distance in neural activity between two images presented in the same perceptual stage, the pre-disambiguation black and white pictures, or the same picture shown post-disambiguation, or the corresponding grayscale pictures. And then these three squares are neural distance between two images presented in two different perceptual stages. For example, this diagonal would be between the same image presented pre and post. And here is the result we get from V1. The first pattern you see here is that this square is redder than this square. What that means is that neural distance between different images, different black and white pictures presented post-learning is larger than pre-learning. And the second thing you see is that there are two blue diagonals here and here, but not here. What that means is that the same black and white picture presented pre and post-learning are represented similarly. No surprise, big one represents image features. But also you see this blue diagonal, but not here. That means that after the one-shot perceptual learning, neural activity patterns in V1 shift towards the matching grayscale picture. And all of these effects are highly significant, as shown here. So the big surprise came when we opened up our microscope and we looked outside of the visual regions into frontal-parietal regions, frontal-parietal network shown in red here, and the default mode network shown in green here. These are regions traditionally ignored by vision science researchers, and they are not thought to be involved in visual tasks, a basic perceptual task like this. But what we found is that we found the same patterns here and even stronger. For example, in posterior cingulate cortex, some may say the pinnacle of cortical hierarchy furthest away from sensory input. We have a pattern like this, and to visualize it, we can take the images we have, and which 33 black and white pictures presented pre-learning and post-learning, and they're matching grayscale pictures, and project them into this two-dimensional space where the distance between dots represent the neural distance in this region. What you can see is that pre-learning, the black and white pictures, different pictures are all represented very similarly. After learning, they are represented distinctly and more similarly to the matching grayscale picture. Okay, so it's a very large-scale brain network phenomenon. And a puzzle in this field, a long-standing puzzle, is that reaction times are very slow. They're not like your typical object categorization tasks where you might get a reaction time around 300 milliseconds. Here we found that even post-learning for recognized black and white pictures, reaction time is at the order of 800 milliseconds. So what is the neurodynamics underlying this phenomenon? Using MEG and whole brain decoding, we asked how does the neural distance between these black and white pictures pre versus post learning, and between the picture and its matching grayscale picture change over time. So first, let's look at the neural distance between a pre-disambiguation black white picture and its matching grayscale picture. Different pictures, and because here you don't have the benefit of learning, you see meaningless patterns, and here you see a crab. And what we find is that neural distance between them shoots up after image onset and stays high during the 2-second image presentation. And if we compare between the black and white pictures presented before and after learning, we find that the neural distance between them ramps up and reaches significance at 500 milliseconds. And if we compare between these two, that's a black and white picture post-learning and the grayscale picture where the image sensory input is different. But here, because you have the benefit of crowd knowledge, you have similar recognition content. And you find that as far as large-scale brain dynamics goes, they are well distinguishable within the first 500 milliseconds. And after that, they are indistinguishable. And this kind of slow dynamics tells us that it really requires recurrent neurodynamics across large-scale brain networks, not just the ventral stream, but also frontal-parietal regions to solve this task. Okay, so this basically summarizes what I just told you. And a more recent paper from our lab digs in a bit more into the mechanisms involved underlying these fMRI patterns. Okay, so next, let me go into spontaneous brain activity. It's well known that spontaneous activity is very prevalent in the brain. It explains 80% of energy expenditure in the brain, more than 80%. And it's got these rich structures that are highly relevant clinically. These rich spatial temporal structures of spontaneous activity breaks down in different neuropsychiatric disorders. And we have good models about these resting state networks in computational neuroscience, but yet we don't yet have any AI or computable models that incorporates realistic spontaneous brain activity structures, to my knowledge. And I think that's going to be a very interesting field to watch out for. So what I told you in the earlier part of the talk is that connectivity patterns that encode past memories provides one source of prior knowledge that can strengthen your perception. It has proposed that spontaneous brain activity provides another source of prior knowledge. The idea is that spontaneous activity fluctuations might sample the prior distribution, which constrains the posterior distribution. And an alternative hypothesis in the field that has also been very prominent is that spontaneous activity may have nothing to do with sampling of distributions. It may reflect mainly behavior of arousal state changes. So we sought to test these two hypotheses by asking, does spontaneous activity influence perception in a content-specific manner, which would be consistent with sampling theory, or in a non-content specific manner, which would be consistent with the idea that spontaneous activity reflects behavior or arousal state changes. And we also wanted to know how exactly spontaneous activity influences perception. Two alternatives are that it could influence perception by changing sensitivity, by changing your perceptual sensitivity, which would suggest that there is a signal to noise ratio change, or it could influence your perceptual decision criterion with no change in SNR. We probe this question by presenting human subjects with stimuli that are titrated to perceptual threshold. It's a paradigm that has been used previously to study access consciousness, related to the previous talk. So we titrate the stimulus contrast such that when the same stimulus is repeatedly presented, the same subject would sometimes recognize the content of the image, and other times they will tell you, I see only meaningless noise. I don't see any pattern here. And that correspond to hit and miss trials to get a sense of a particular individual's baseline tendency to say, I recognize a meaningful content. And this is related to Marta's talk earlier. We also presented them with these scrambled noise pictures. And in this case, if they answer, I see a meaningful content, this would be a false alarm or false perception. If they say no, it would be correct rejection. These scrambled noise pictures are phase scrambled, so they preserve low-level image features that differ across categories, but they do not contain any meaningful content. They are purely noise. In this task, subjects recognition rate for real images is around 50%. That's because we titrated the image contrast to reach 50% recognition threshold. And for scrambled images, there are about 20% of the trials where they answer, I see a meaningful content. And this would be the false alarm trials. And some might say that it's a model for understanding hallucinations, allowing us to probe similar mechanisms in healthy subjects. So the first hypothesis that spontaneous brain activity might encode behavioral arousal state changes predicts that this activity influences your perception in a non-content specific manner. In other words, when this activity is high, the subject is more likely to recognize this threshold level image, regardless of what's the image input. And if this is the case, that means that we should be able to use pre-stimulus spontaneous activity to predict participants' recognition outcome that's reported seconds later. And indeed, we were able to do that using pre-stimulus MEG activity. We could predict participants' recognition outcome, but moreover, this is, I should say, a linear decoder, logistic regression decoder. Moreover, when the decoder was trained using stimuli from one category, it cross-generalizes to stimuli using a different, from a different category, suggesting that this spontaneous activity influences recognition in a really non-content specific non-content specific manner consistent with the behavior arousal state change theory. The second hypothesis suggests that spontaneous activity might sample the prior distribution. And if this hypothesis is correct, that would mean that there are different spontaneous activity patterns that promote recognition for different stimulus categories. For example, if this spontaneous pattern represented by the blue trace is high, then when the stimulus, if the stimulus is a house, then the subject is more likely to recognize it. But when it's low, subjects would be less likely to recognize the house, and there would be a different pattern that specifically promotes your recognition of animals. And if this hypothesis is correct, what that means is that we should be able to use pre-stimulus activity to predict the stimulus category on recognized trials. So if we only take the yes trials, then the pre-stimulus activity should tell us about the stimulus category, even though in the experiment, the stimulus category was randomly drawn on every trial. And indeed, this hypothesis was correct. We could use pre-stimulus activity to predict stimulus category on yes trials, but not on no trials. So to answer the second question I laid out, how does spontaneous activity influence recognition? A very influential theory in psychology tells us that once we sort the trials into hate miss for the real image trials and false alarm and for the correct false alarm and correct rejection for the scrambled image trials, then we can use signal detection theory to ask, does the spontaneous activity influence recognition by shifting criterion or changing sensitivity? I'm sorry, the animation that is not working. So for criterion, it would be the criterion shift to the left, and for sensitivity, it would be that the distribution between signal and noise becomes larger. So we found that the non-content specific spontaneous activity influences perceptual recognition specifically by shifting criterion, such that when this spontaneous activity is high, the criterion is more liberal. It shifts over here, and subjects are more likely to answer both hates and false alarms. Whereas for the content-specific spontaneous activity, when it's high, the distance between the noise distribution and the signal distribution becomes enlarged, and it specifically enhances participants' perceptual sensitivity. So now we have a candidate spontaneous activity process that we think may reflect behavioral arousal state changes. We can test that directly because it's known that moment-to-moment pupil fluctuations reflect changes in cholinergic and norepinephrine signals, that's widespread projections to the cortex. In animal models, it's known that when the pupil diameter is larger, the locomotion activity in rodent models is higher, and membrane potentials recorded intracellularly from sensory cortex is higher, meaning neurons are more excited. And we recorded real-time pupil diameter in this task, and we found that the non-specific spontaneous activity correlates with the trial-to-trial changes in pupil diameter, but not the content-specific process. Okay, so to summarize, what we found here is that both theories are correct, and there are different spontaneous activity processes that are consistent with sampling or consistent with behavioral arousal state changes. In the sampling of the content-specific spontaneous process, it constrains your perceptual interpretation, and it enhances perceptual sensitivity, whereas a non-content specific spontaneous process correlates with arousal changes, and it shifts participants' perceptual decision-making criterion. Okay, so lastly, I'd like to tell you a little bit about top-down inference we've found. So firstly, so let's take a closer look at these false alarm trials. So in these trials, we also asked participants to answer which category they saw. So if you saw a meaningful content, even though the sensory input is noise, what did you see? And we found that their reported category matched with the category that the scrambled noise image was generated from. So remember, I told you that low-level image feature, the scrambled images preserve low-level image features of the real images. So that suggests that these trials are really false perception, genuine false perception trials, not just the bottom press mistake. So we wanted to understand more what happens in these false perception trials. And here we went back to 70 fMRI, and we found that if we contrast the real image trials and the scrambled image trials, there are specific high-level brain regions that are more involved in the scrambled image trials shown here. So the blue regions are activated in this task, inferior frontal junction, and this is a region that was also mentioned in Jim's talk yesterday, and also anterior cingulate cortex. There has been some very good rodent work about how ACC can send top-down templates to V1. And so these regions are activated in this task and even more activated when the image input is scrambled noise. And the orange regions here are part of the default mode network. They are deactivated, and they are deactivated more when sensory input is noise. So that's just activation. We want you to understand where in the brain can we find information that allows us to tell what the subject is really perceiving. So we decoded perceptual content using multivariate voxel patterns in different regions. So this picture shows the activated and deactivated brain regions in this task. And to our surprise, we found that we could decode perceptual content in the scrambled noise image trials from pretty much all of the activated regions shown in orange and deactivated regions shown in blue here, but not in subcortical brain regions. And this pattern is very much reminiscent of the recognized trials for real image trials shown in blue here. So every time there's a circle around the dot, it means that the decoding is significant above chance. But for real images that are unrecognized, we can't decode image category in any of these regions. What this says is that when sensory input is especially ambiguous, it only contains noise, the higher order, very higher order brain regions in front of parietal networks are especially engaged. And both in the activated and deactivated brain regions, there is information content that directly relates to participants' perceptual outcome. Okay, so now no talk about spontaneous brain activity and perception, in my view, is complete without considering this really interesting psychophysical phenomenon, bistable perception. So these are images where the ambiguity is artificially high, such that the two interpretations, or sometimes more than two, they compete each other. And when you focus your eye in the center of the image, the perception outcome of the image can't help but flip back and forth between the two different interpretations. So, for example, this one can be seen as a woman walking a man's face. And they have intrigued psychologists for over 100 years. Many years ago, we published a study where we simply used the searchlight across the whole brain to decode perceptual content for the bistable images shown in teal here, and for similar images that are where the ambiguity has been removed, shown in green here. And we found that when the image is not ambiguous, we can decode perceptual content in visual regions, shown in green here, and the dark blue, which is the overlap between the two, for example, V1 and IT regions. But when the image ambiguity is high, we could decode the perceptual content additionally in frontal regions, including ventral frontal cortex and orbital frontal cortex. So this provided a hypothesis that when sensory input is especially ambiguous, then higher order regions send a top-down template to facilitate perceptual resolution. And another interesting finding here related to lifelong priors is that the perception of these images are not really symmetrical. So in this experiment, we showed people these bistable images, but this top-down view where you are looking down on the cube is typically more prevalent in perception than the bottom-up view. That's because we have the lifelong experiences of seeing cubes on the ground that hang up in the air. And we were lucky to record intracranial EEG signals from neurosurgical patients who perform this task. And we found that when participants perceive this view, the more prevalent view, there are more top-down input information flow across large-scale brain networks from frontal to temporal region, from temporal region to occipital regions. That's shown in blue here, blue-purple color. And when they perceive the alternative, less prevalent view, that is more bottom-up activity shown in red here. For example, in the ventral stream, the preferred view is corresponds to much more top-down input. We put together a very abstract, simple mean field model that basically incorporates hierarchical predictive coding and attractor network elements, where there's a top bias that is constant in the play, and then each lower level region in the network computes a prediction error which propagates forward. And the model captured both the behavior phenomenon and the neural phenomenon here. That suggests that top-down input in the brain can carry information related to lifelong prior knowledge to constrain our perception. So this is the last part of the talk where I showed you that for false perceptions, there is heightened involvement of higher order regions suggestive of top-down inference, and top-down input can carry long-term priors. Okay, so to summarize, to quote Richard Gregory, perception is solving puzzles, and perception is the inference about the cause of sensory input. I showed you today about how memories from one-shot learning can shape perceptions through evoking large-scale recurrent cortical processing, how spontaneous brain activity can shape perception through two different mechanisms, content-specific and non-content-specific. And lastly, I give you some concrete examples of how top-down input can carry predictive information that generates false perceptions and shape our perception when sensory ambiguity is high. So I like to stop by thanking all the people who did the work that are the names shown in bold here and my funding sources. Thank you for your attention.
Wonderful. Thank you so much for a really interesting talk. So we're going to skip the break after this talk in order to try to get ourselves back a little bit on schedule, but I would like to take time to just do at least one or two questions. So the most upvoted one is from Paul Shizek. He asks, the computational problems you've discussed, inference, ambiguity, prediction, etc., are applicable to the kinds of processing that take place in the ventral stream. Do you think that similar computational problems arise in dorsal stream processing? If so, do you think the mechanisms for dealing with them are similar?
I would think so. I talked about memories, spontaneous activity, and top-down inference as sources of prior knowledge. So I think at least memories, at least the top-down inference and spontaneous activity would be common to the dorsal stream. And then in terms of the one-shot perceptual learning task I showed, there we have beautiful neural imaging correlate data and lesion data that tell us the changes in large-scale activity patterns in this task, and that the task does not depend on the hippocampus. But where exactly it is in the cortex, we don't know yet. By combining MEG and fMRI data in cross-model RSA analysis, we find that the recognition-related activity onsets first in IP cortex and frontal parietal regions almost simultaneously. So it kind of constrains the location to be there, but we don't yet know exactly where. But to summarize, I think I would expect at least similar spontaneous activity and top-down inference findings in the dorsal stream.
Great. One more quick question for you then. Regarding the black and white photos that are recognizable after seeing the full photo, this is from Andrea. I am wondering how we can say this is one-shot learning when most people have seen many cats or dogs, to take the examples of the photos before. So seeing this particular photo that fills in the photo, isn't this really a matter of consolidating on priors as opposed to one-shot learning?
I would say that it completely fits one-shot learning definition in the sense that this picture, this black and white picture, you can present it to a participant and they can study it, for a long time. and they don't have that perceptual closure. They don't see, the content in the picture. But then, in the laboratory, we can, all, in the real world, you can show people a matching picture that's, ambiguous. And all of a sudden, the black and white picture, when you show them that picture again, and, even many months later, they recognize it automatically and effortlessly. So I think it, if that's not one-shot learning, I'm not sure what it is, what one-shot learning is. And then to come back to the lifelong prior, so there is a lot of individual heterogeneity here, where some pictures will be, impossible to recognize for some people, and recognized right away for others. So I think in the second context, of course, that's not one-shot learning, because exactly like you said, in that case, you are bringing lifelong knowledge about cat or dog to solve that picture.
Okay, great. Thank you very much. Well, let's thank our speaker again for a great talk. And now, like I said, we're going to skip the break. So we're going to move directly and hopefully as quickly as we can on to our next speaker, Alison Gopnik. We'll be back with you in just a moment.